The overall goal of this procedure is to investigate the function of extracellular vesicles released by the malaria parasite-infected red blood cells.
This method can help answer key questions in the malaria field, such as how the parasites regulate parasite-host communication. The main advantage of this technique is the visualization of vesicular uptake by endothelial cells and the study of the regulatory function of the vesicular RNA by endothelial cells. Demonstrating the procedure will be María Hernández, Smart Mbagwu, and Babatunde, all grad students from my laboratory.
In a microcentrifuge tube, add 100 micrograms of extracellular vesicles and 1 milliliter of phosphate buffered saline. Then centrifuge the extracellular vesicles at a maximum speed of 20,000 times G for 7 minutes to form a pellet. Once the centrifugation is over, discard the supernatant without disturbing the pellet. Then, dissolve the extracellular vesicle pellet in 100 microliters of diluent C. To ensure complete mixing, pipette it several times. Next, in a new microcentrifuge tube, add 4 microliters of the pKh67 ethanolic dye solution to 100 microliters of diluent C. Then, vortex the mixture well. Prepare the 40 micromolar 2x pKh67 dye solution just before the staining process. Then, quickly combine 100 microliters of 2x extracellular vesicular suspension.